This is a production of Cornell University.
Thanks for the invitation. I'm very looking forward to share some thoughts and what I'm doing or what I'm thinking to do at the University of Florida, and thanks for the opportunities to collaborate. So maybe a highlight of what I'm trying to do and develop here at UF based on what I done in Corteva and build on that foundation is to reimagine agriculture as a solution to climate change. The narrative today is the other way around, but I think there's a huge opportunity and it's quite imperative that we do. Of course, I'm not going to be the one to do it. Hopefully I can make a contribution to it. Now, more specifically, I'm looking forward to develop and apply breeding technologies. UF brings a lot of opportunities to think about. I think we have like 50 breeding programs that will expose challenges and help us develop methods and create opportunities to think about the circular bioeconomy. There's a tremendous opportunity here to work with Ed's group on the Circa project, and I'm looking forward to that work too. And finally, something that caught me a little bit by surprise, I was behind the iron curtains and a little bit detached on climate change breeding, if you will, and more focused on the shorter term. And when I start doing some research about where we were at, I found I think there's a big opportunity here to inform and collaborate to put a framework together on how we're going to do it. It was surprising that the whole or the opportunity I think we have in front of us. I think the narrative was mainly driven by agronomists rather than breeders. So anyways, I think this is an opportunity to have an impact not only as Florida, but as a community of breeders, in particular working with climatologists. So I'd like to start with what problem we are trying to solve. We're talking about climate change and circular economies. And to me, is this high throughput, high externalities agriculture is no, it's never been too sustainable, but it's certainly not sustainable right now. An eye-opener for me was to read a report from Rockefeller Foundation, and the two numbers that are really striking are the agricultural economy generated $1 trillion in value and generates $2 trillion costs in externalities tied to human health and environmental impact. So today, the way this high throughput, high externalities agriculture is working is generating more problems than it solves. Of course, we need to eat, so it's a kind of a difficult problem we have in hands. However, I don't think there's a limitation to change how things are done. And I will build a little bit of how we got here based on this grain maize example. What we would see in this graph is the trajectory about 100 years of plus of breeding, where for most part, it was all about keeping the hybrid standing, mechanization, increasing the throughput of the system, right? That was the double cross area. Keep those plants standing, plant more acres, and leverage the capacity of corn to deliver on that photosynthesis. In the single cross area is sort of is synchronous with the increasing use of fertilizers, irrigation. Essentially, we're breeding for plants that were transforming increasingly amount of inputs into outputs without regard to what was going on with the aquifers and whatnot. In the last part of the biotech, I think we start getting into that circularity by reducing the amount of pesticides but still it was in the same path to create these machines that transform more and more inputs into outputs. It wasn't until that we worked with the ACOMAX drought tolerant, looking at increasing use efficiencies of water and resources, that you can start seeing an incipient or the onset of some circularity thinking that is built into the breeding programs. So by large, the breeding has happened without regards to externalities, if you will. It was pretty much one-dimensional. So an alternative paradigm that is quite appealing is the circularity. I came familiar with this as MacArthur Foundation started providing. It's really intuitive, this nice cartoon, get everything within the cities, recycling as a concept is great. I think the principles will help to bring to practice some of the sustainability ambitions. The industrial ecology framework is quite practical in terms of engineering, something that is kind of hidden, and always I wonder why it was in there. You know, is the intention of the MacArthur Foundation. I think healthier food product is always a good thing, but it wasn't clear to me how that has anything to do with circularity. And to understand, I start scribbling a little bit in how the different elements, or at very high level, relate to each other between climate change, human health, and agriculture, essentially to understand to myself how these things connect to each other. And the clear outcome of this sort of a system that reinforces itself is, if we keep in this path of high externalities agriculture, we are going to have impacts on the climate. It will have a negative impact on agriculture, and this is going to spiral down. Same thing with the externalities, poor human health, reduced productivity. So what do? We put more inputs to compensate for lower inefficiency in agriculture, and we keep things, making things worse and worse. So this thing is going to spiral down until we break the whole system. So how you sort of disrupt this vicious cycle? And here is where it's sort of a click, that sort of little corner in yellow that you barely see in terms of human health and diet is the signal between what humans want from agriculture to deliver. If we want cheap beef produced with large quantities from Ogallala, Nebraska is going to continue to produce corn. That's what society is demanding. So diet essentially is at the core of how we are going to signal agriculture to shift, and how we are going to think about agriculture in different processes in terms of whether it's ethanol or beef produced with a reasonable amount of protein from different sources, or plant-based agriculture. I don't know what is the answer, but certainly what connects and what can disrupt this cycle is this diet as we shift the purpose of agriculture, the objectives, and how we reshape the bioeconomy. So my work in hypothesis is plant breeding, emergency engineering, and certainly artificial intelligence. Is a mouthful worth of words. Hopefully it will become a little more clear what I'm thinking throughout the presentation, is what is going to enable to create the products to reimagine the agriculture. Essentially, plant breeding is what, if diet is going to signal agriculture what to do, plant breeding is that mechanism or methods we have to create the products for the farmers to re-engage or reimagine this agriculture and make it a reality. So with that, then the question is how we are going to transition. Let's assume we can do it breeding. We know how we operate, how we go from linear to circular agriculture. And to me, has a lot to do with being really deliberate, and to be deliberate, we need to engage design thinking. And going back to my first statement of we were fueling inputs to corn and ask for outputs and increasing amount of outputs without concern of water quality, what that is representing, that G by M technology and multiple traits, we were 100 years of one dimension, essentially, is in produce and vegetables, maybe quality, in corn, maybe yield, maybe morphology, physiology, if you want to start getting into more efficiencies in terms of how we are going to increase yields. But nobody pay attention into that selection index. So what was the nitrogen emissions? Is it water quality? How much water were we pumping from the Ogallala? Or are we sequestering carbon, or what are the emissions? So to transition to this circular bioeconomy, we need to start thinking differently on how we're going to create these selection indices, if you will, and how we're going to generate the information. We can't really wait 30 years for a data point to come back on what is the carbon sequestration. So here is where I think modeling and simulation, it can become quite critical. Same thing with nitrogen, a little bit less on water. So essentially we need to develop methods to empower decision makers to approach some of these dilemmas. Sometimes is water and productivity is going to trade against each other, so we need to make a decision. The other is a shift in mindset, where we try to put simple solutions or provide simple answers instead of thinking about simple methods to manage complex systems. Think about regulations on nitrogen, papers recently published on how are you going to tax nitrogen. That's a really bad idea. Everybody is going to make the wrong decision instead of creating frameworks that enable everybody to make the right decision, yet attain the goals of reducing externalities. So how a crop design framework might start looking like, or what are the components that at least I think we need to bring to place? And the first one is start thinking about processes rather than states, and how we think about the phenomics and all the engineering that goes with it, to start phenotyping or conduct phenotyping that leads to enhanced scientific and genomic understanding, because that is what is going to enable us to predict not only yield, but all these other consequences at the system level of, if you make a decision, breeding decision, how much is going to impact yield, water use efficiencies, nitrogen, carbon sequestration. You can run a simulation for 30 years for that matter. So we empower the decision maker and provide information that otherwise is not going to be available to them, certainly not today. And what brings everything together is, in my mind, is artificial intelligence, and I will elaborate a little bit more on that. The other piece, if we're going to design things, we need to measure success. And to me, a very simple way to measure success is through genetic gain. But the very single identity here is that genetic gain has to be greater or equal to the amount from the growing population, whatever that is coming from. And while I'm not ready to put or calculate the time derivative here, I think this identity is quite useful to think about what is the problem with increasing genetic gain, and the equation right there, and the heritability is a function of the variation or genetic variation and divided by the G by interactions and the errors. Again, I know some breeders in the audience, this is trivial. Hopefully for the climatologists is something that become a little bit more useful to understand, that the environment is one of the problems as we interact with the genetics to hamper our abilities to make genetic improvement and improve crops. So this is an example that we can go through, that G by E, it's an emergent property that depends not only on the genetics, but on the expression of the physiology and the range of environments that constitute that population of environments we are trying to breed for. On the left, you would see, hopefully, a number of, in the vertical, a number of different types of G by E, starting on the top with no G by E. It's a very simple representation. You have two environments on the X, yield or any property on the Y. Parallel lines means you're making genetic gain. Whatever decision you make is the right one, whatever the experiment you conducted. The second from the top, you'll see that is heterogeneity of variances. So yeah, there's a G by E, but doesn't affect your decisions. Essentially, in one environment, one hybrid is going to eat more than in the other. The lack of correlation is here the devil that we need to fight all the time. And when things combined, the things can get pretty ugly. And that's probably some of the drought examples we can elaborate. So if we start on the figure on the center, look at the bottom, we have a yield scale. Just want to bring to attention, this was created by Randy Clark. This was Susan's student from Cornell. And the Y is each of different traits, different colors here. It will represent a trait. And on the Y, it will tell you how important that trait is to explain variation among different hybrids at these different levels of yield. So if you take this yellow trait is silk emergence rate, how it responds to water deficit, you can see if environment 1 is at, let's say, 600 grams per square meter, probably flowering stress, a hybrid that has a good silk emergence out yield the one that it doesn't, explain a lot of variation. And as you increase the water availability, that trait become less relevant, will explain less variation, become even inconsequential at 1200 grams per square meter. Environment 1, environment 2, variation that trait will lead to heterogeneous variance G by E, but certainly the decisions that you're going to make is not going to be affected. Think about the other one is canopy size. Canopy size is related to light interception, carbon assimilation, transpiration. This is what's been driving yield in corn for the last 100 years in terms of either plant or a canopy level. So if environment 2 is an irrigated in Nebraska, big plants consume a lot of water, it's a great idea. Your pivot breaks, your environment 2 becomes environment 1, big plants consume a lot of water, enter into drought, and those plants will crash. A lot of the Aquamax drought-tolerant material has some of these characteristics of conserving water to prevent that crash. So this trait, depending on the environment you're looking at, it will generate that lack of correlation and will trade, if you will, the performance on one environment against each other. And if you combine these two traits, the silk and the canopy size, for example, in environment 2, you have a big plant with poor silking abilities, is going to have heterogeneous variancy because it's going to crush really miserable, and you will see some pictures throughout the presentation. And of course, it's not going to get the silks out. And on the other end, it is going to produce yields, but not as much as it could. So again, depending on the trait combination, depending on which environments are you sampling, you are going to see different patterns of G by E. So the argument, or my argument, is that to enable to manage this complexity and emergent patterns, particularly in genotype by environment interactions, and think about the environment to put the management there too, we need some biostatistical machinery to be able to predict this. While this appealing to think about deep learning and some of these tools, I think by harnessing the scientific understanding and coding everything we know, and we call that symbolic AI, with the statistical learning capabilities, whether it's deep learning or something else, call it statistical learning in general, as a form of sub-symbolic AI, we are going to make much progress. There are some papers published recently, they call the future is hybrid. Carla Gomez applied this to material sciences. We got to the same path through looking at genotype by environment interaction. So you can start seeing from remote sensing, material science, genetics, we are sort of coming to the same conclusion that harnessing scientific understanding is a good idea rather than put data in a black box and hope for the best. So if you think about the phenotype, plant phenotype, crop phenotype as a function, and T is for total, so that's your theory of everything. You can break this in what we know, that's your scientific understanding, what you don't know, and you would like to figure out. At some point in time, we didn't know about photosynthesis. Now we know. So essentially, you start moving knowledge from, or what you uncover from H, and put it into Q as a detective. This is pretty trivial, but I just want to make it formal how crop models map into this Q. This is what we know essentially. So out of the FT, which is the world that's around us. And crop models, as this introduction, are formal systems that represent the knowledge on physiology, environmental science, and more important is a cognitive model because it represents the topology of the network, how elements or components of the system are causally related to each other. Just you might have heard me talk about this other times, but remember this network, remember this cognitive model, because you will see how when you start combining symbolic and sub-symbolic AI, the same way genomic prediction borrow information, genetic information to make prediction about individuals that were not tested. We can borrow information through these biological networks to make predictions for even genetics that are not were tested. So it's a different way to borrow information to improve the prediction of algorithms. So let's call crop models or symbolic AI. Do these models work? And one way, at least, is important to me is the ability to predict importance of traits, but certainly emerging patterns of G by E. You saw the work done by Randy. This is a different example now at a larger scale. We start doing these experiments on limited transpiration or conductance response to humidity with Tom Sinclair to ask the question, is the trait like this even remotely relevant to undertake further research? And it was, and that motivated a lot of investment within Corteva. What you can see here, look at the regions A and B on the Corn Belt, the Western Corn Belt, this genotype 1 out yield genotype 2 in dry years, panel C, or even wet years on panel A. Drought is pretty generic and generalized there. With some assumptions, you may even think about if there's no G by E or maybe some heterogeneity or variances. Problem comes when you start going to region C, where in dry years it's going to outperform genotype 2, and now you have the crossover interaction and lack of correlation. So a relatively simple model and a trait that even at this point, well, at this point, but a little bit earlier was a hypothetical one, we were able to start detecting what were maybe the consequences and what were the regions where these traits may be useful. So this is a part of the symbolic AI that makes us comfortable. Yes, these models can capture a lot of behavior based on our scientific understanding, even a scientific speculation of functioning traits. So now that you have these crop growth models, would like to bring to your attention is there are things that we know within the crop model, like use efficiency in photosynthesis, and some things that we know, maybe partitioning, but we either don't want to measure, or it will be difficult or too expensive, and we want to estimate. So think about these crop growth models. We have a vector of known parameters, unknown parameters we want to estimate, and of course you have the forcing through environment and management. Just want to use that to introduce this integration of symbolic and sub-symbolic AI through CGM-WGP, which is crop growth model whole genome prediction. Think about the crop growth model as a way to an algorithm to generate data, not field data, it will be in silico data, and that will be a function, again, of a vector of parameters and a forcing, and the whole genome prediction is going to be an algorithm that in this case it's a linear mixed model. The animal model, more specific, will generate the ability to use marker or genetic or pedigree information, depending on what you're using, to estimate a mean and the vector of, let's say, marker values. So essentially, think about you have now a function that can tell you tell you something that you can use to predict any particular trait. In this case, Y would be the RUE. If you measure, you train, you'll be able to estimate that number, and you can use within the crop growth model. So when you combine these two things, you can put together a likelihood function, which gives you that probability of, given the data that either generated or observed, what is the probability of the markers given the data you observed. I say it the other way around. Sorry, in the consequence-based theorem, the posterior will give you the probability of the estimated marker effects given the data you have. And more important, I think that we don't say explicitly, is conditioned upon also of everything else you assume remain constant. For example, if you have a very complex crop growth model, you may have 40 parameters. You're estimating RUE is essentially 1 out of 40, that this posterior distribution of RUE or marker effects for yield and RUE, if you will, is going to be conditioned upon all that from all the parameters you assume to remain constant. Again, I think this is important to bring to your attention as you start thinking about using some of these methodologies. So an ABC approximation computation study, I just used this in class here at University of Florida to teach a plant breeding student. I use a tight prior distribution for radiation use efficiency, conversion of light into carbon. So it converges within a reasonable amount of time. So think about, we sample this prior, we use the linear mix model to estimate a genomic breeding value. Then with those genomic breeding values, we simulate a multi-environment trial with parameters we know, plus the RUE, given a set of environments that we sample. We calculate our simulated data set. We compare the true with the observation with the simulations. If that delta is acceptable, we keep it to build our posterior distribution and estimate the marker effect. If not, we discard and we continue going through this process over and over. This is slightly different from what we've done with Frank Techno in the past, but I like the way it connects more explicitly and clear the crop growth model with a genomic prediction. So now we can run a simulation to see if we can recover the truth. Here, what I created is a map of yield for a very simple multi-environment trial of 2 environments. Each point is a different genotype, that the only difference it has with respect to all the other genotypes is the value of RUE. And you can see there's a bifurcation at about RUE larger than 1.45. You can see that there's a split in the behavior in environment 1 and 2. Many times these environments, when we use for a GBLAB, we just combine them, make a prediction. So we don't have the capability to discriminate unless we have a very compelling evidence that these are 2 environments to belong to different environment types. So when we run the algorithm I just described, you can see that we can recover the RUE very nicely. The posterior, you can compare the posterior and the prior. But you can see here is there's a shrinkage. You can see the true RUE, which we defined in our multi-environment trial, run between 1.2 and 2. And the predictions is 1.5 and 1.65, and this is because of the shrinkage that it was imposed when we calculate the linear mix model. But this is important, or is this important to make prediction and rank hybrids? And what you can see here is not. Essentially, the algorithm shrunk the range of RUE, but still, in a condensed manner, we're able to estimate all the rankings accurately for both environments. And what's important to note here, in contrast to the GBLAB, we would not calculate this value for one environment and the other one. We just feed all the information and the algorithm figure out how to is going to partition that information, environmental and genetic, to make the predictions and estimate the performance for the two different environment types. When you don't know the environments, these become actually quite powerful. So does it work? And rather than tell you exactly what we did in different cases, certainly I'm more than happy to refer to different papers and you see exactly how the experiments were conducted. I would like to take the opportunity to reflect on how I would do it if I need to apply this to a cassava in other places, or sorghum, or how I'm thinking about applying this in eggplants or other vegetable crops for which we don't have 10 years ahead of us to develop crop models or the funding or the scale of a breeding program for maize at industrial scale. And to me, the algorithm is pretty simple. Start very simple models. It doesn't have to be overcomplicated, but look at combining processes for which you have well-defined constants and focus on the traits that the breeder is telling you that is varying on the germplasm, or look at the hypotheses that you can formulate about what traits may be varying on the germplasm. Then you can leverage existing data sets. There are a number of breeding trials of high quality. There are a lot of physiological studies can be conducted. And within a Bayesian framework, you can accommodate all this uncertainty. And then develop your CGM WGP through iteration. Just don't wait until you get the perfect crop model or genomic prediction algorithm. Start iterating and ask the question whether the genotype by environment by management system of your interest is this technology and methodology can give you an edge relative to alternative approaches. And then you start applying within the breeding program, because that's where it's going to surface all the opportunities to improve and start guiding the development of the method. And of course, all this is hypothetical up to this point, so you can start designing experiments and put your effort and resources to test these hypotheses rather than to start building part of the background. So as I mentioned, if you want to learn about exactly the methods and how we did it, we published this, a lot of this with Christine Dippenbrook, another graduate from Cornell, when we were in both at Corteva. This particular case was a very large half diallel experiment, include about 2,300 hybrids. I think it was just one tester in this particular case, and run about 23 locations that which evapotranspiration range for about 800 to a hair below 400 millimeters. So that tells you a little bit about the range of environment. And as you saw in the image I Randy Clark created about the yield performance and the different traits, you can see on this more harsher environment, the G by E increases, a lot of associated with the silk elongation. These are not extreme environments where you expect that nothing is going to work, right? Everything is dead. So there's a sweet spot on how this accuracy difference between the crop model whole genome prediction, which has that biostatistical machinery to predict G by E, compare against, let's say, base A or any of the genomic algorithms that does not have that machinery to predict G by E. So you can see in well-water environment, everything is well water, so it doesn't really matter. There's not a lot of G by E, so not surprising, the accuracy difference is very low. I started pushing this to more stress environments where the G by E and the trading between water, carbon, water conservation and whatnot. You saw a couple of examples of that with the limited transpiration and the silking. You can see any biostatistical machinery that can capture that behavior is going to have an edge over an algorithm that it doesn't. And this is what you see in this figure. As that G by E increases, the accuracy difference increases. This is a more recent paper we put together that used a bigger data set, less diverse, I think, I would say, but include about 7,600 hybrids, about 6 years of experiments in the Corn Belt. The range of evapotranspiration is large, is from 250 to 800 millimeters. And we generate, and this is an images for the checks between total barrenness and pretty much half full years. The goal here was to advance the methodology now to train this crop growth model, whole genome prediction, not just on yield, but how we start utilizing other traits to improve the predictive skill of the algorithm. So here, this complex figure, let me take a few minutes here to explain. What you will see in figure A is the same figure you might have seen in other papers or I showed you before. Essentially, you have the accuracy of the whole genome prediction algorithm for yield, trained for yield, and the accuracy of the crop model whole genome prediction. So essentially, you have a whole genome prediction without a component to predict G by E, and whole genome prediction with an addition of machinery, the symbolic AI machinery to predict G by E. And not surprising, particularly look at the accuracy levels that we are dealing with in here, anywhere between 0.3 and negative 0.3 for the whole genome prediction by say in this particular case. There's a fair amount of G by E in here. You can see that by large, most of the time, when you add the scientific understanding, you increase the prediction accuracy. So now move to shed, which is panel B. What we are trying to do here, and you need to read this panel slightly different, because we are trying to predict shed using grain yield information. And the accuracy of the WGP is, well, of course, you cannot do that. So what you are doing is just providing a metric of what would be the predictability if you have that shed data. And if you look at the distribution of all the points, the accuracy of the CGM WGP is pretty much centered around 0. It tells you it can't do it. It's just by chance it generates accuracy of 0.3 and equally likely with minus 0.03. But look, now move to the silk. So we are still training the algorithm using grain yield data alone. The algorithm have no information to predict silk. And the expectation based on WGP should do something that maybe a mean around 0.3 or 0.4. But compare with the shed, the distribution of that accuracies for silk are certainly shifted relative to the shed distribution. So what is happening here is the biological network, that crop growth model, essentially is linking yield to silk to kernel numbers. So there's a path, and as I mentioned, that this scientific understanding help you build this causal relation and topology. So call it symbolic, sub-symbolic AI, what it's doing is leveraging that topology of that biological network to borrow information from yield to be able to say something about the silk. In the genomic prediction algorithms, what they're using is markers and pedigree relationships to borrow information from the different relatives to make prediction of something we didn't observe. In this particular case, we didn't observe silk, but we are predicting it, borrowing information which is encapsulated in that biological network. So think about those two algorithms that are using exactly the same strategy to borrow information to say something about a trait or a relative that we have not tested, or an environment we have not tested. And now if you move to kernels per year, it's a similar situation. It's still borrowing information, probably spillover from silk, to predict something about what's happening in the kernels per year. You have a whole genome prediction algorithm for kernels per year without having to phenotype kernels per year at all. Again, the accuracy is not great, but you can see certainly a shift on that distribution, which is looks like different from 0. I wish I tested that. So now you go to the panels on the lower set of panels. Now what we are going to do is we are going to predict grain yield, but we are going to use data from grain yield, shed, silks, all together. We haven't used the kernels per year data in any of these situations. So you can see that grain yield, I think you can do a good job, yet now that we have shed and silk, the causality and these predictions are more robust. For shed, now that the algorithm has information about shed, has no problem making that prediction. You can see the shift in the clouds of points, either above the one-to-one line or at the same level of the WGP. And same thing with the silks. Essentially, it maintains that edge. So you can see the point above the one-to-one line, but for all the other points that were not able to borrow that information, perhaps it was more constitutive type of effect on silking, now is able to harness that, leverage that information of phenotypic information. And for kernels per year, you can see a slight improvement, and this gives me to a good segue that this is an iterative model building. Let me reemphasize what I told you before. We start with the simplest model you can, and you find your errors and you iterate. So here, what we've done is, again, building with another graduate. I'm sorry, a graduate from Cornell helping us build this phenotyping platform, and to test the hypothesis that the error that we are seeing in here in the prediction of kernels per year is due to lack of biological processing in the model. So we tested this, we put it in the crop model, we have a good handle on prediction of kernel number based on growth rate. The models can generate these emerging patterns of behavior. So this hypothesis, we can really reject it, but doesn't seem to be the favorable hypothesis in my mind. So we can start thinking about alternatives. And this is a paper that Adrian Roeder, professor in Cornell, lead this nice publication where we advocate that part of the lack of predictability in some of these systems has to do with emerging behaviors that we certainly don't have included in the crop growth model, but it's not just a function you put in there, you need to think a bit harder than that. So part of the argument here is that all these barren plants that may compromise the yield, and that's why we don't have the predictability, is due to these bifurcations, and the higher increase your plant population, the more random this becomes. So what we are working right now is on frameworks that help us not only create this upward causation, again, the crop growth model network that I described, think about an upward causation, all these links from, let's say, trait to yield has a consequence on the these emergent phenotypes, again, the relationship of plant growth and kernel number, but also there's a downward causation from competition, resource availability that need to be incorporated. So again, this is an area of active research that we need to work out to increase the predictability of these models. As I mentioned before, and I will emphasize as many times as possible, there's not a lot of time to wait to have the perfect model. So even with the information we have and the accuracies we have, we can think about using artificial intelligence in the form of the algorithms that I presented to provide solutions for thinking farmers and breeders. This particular case, I think, is about the dashboard that we can present to the grower or the breeders, so they can start assessing how they're going to prioritize food production, water use, and carbon. A very trivial example here, but we have made the case, is we can increase the carbon sequestration or carbon production stored in the dry matter if we increase evapotranspiration. But the question is, if we are going to do that in Nebraska, pumping water from the Ogallala River, that's a very dumb idea. So again, now we have the breeder has the information so they can be deliberate about their decisions. And this give me a little good segue to start thinking about how we're going to breed for climate change. And hopefully we can finish the talk with an example of why I think we are in a very poor situation as we think about breeding for climate change. The case I'm going to use again builds on maize breeding in the United States. That's what we know, and that's where we have a fair amount of data. So the first condition that has to happen is, did the climate change concurrently with the breeding program from Pioneer? And what we see here is different variables. These are aggregates for the Corn Belt. NOAA has a beautiful application that can give you these aggregates of precipitation, temperature, and amplitude. And there's no doubt in my mind when you look at the anomalies relative to the last century, the precipitation looks like it's been increasing. There's no doubt that minimum temperatures increase and the amplitude decrease because of maximums have not changed that much. I think it's been known from Carl in the late 90s, and that trend seems to be holding quite steady by now. So first condition, the climate change during the same period that the breeding program was operating. So now you can start asking questions, how the genetic gain can change with climate change. In the perfect world, we can do is of, let's say from the 1930, which is the beginning of the breeding program, till 1990, we select hybrids that growers decided that they were the best hybrids by buying them. So that can help us select the set of genotypes and monitoring genetic gain. So that is, if you will, is the reference set that we want to consider to estimate genetic gain, let's say 1930 to 1990s. And then what we do, if we feel that the desire is run an experiment from 30 years, 1990 to 2020, to assess the effect of that climate change over the genetic gain. Fortunately, Corteva conducted experiments since 1990 with the same set of genotypes. These are known as the Dubik era studies. Dubik conducted this until the 2000s. Mark Cooper ran those trials till 2016, and when he left Corteva, and I ran those experiments until 2021 when I left Corteva. So we have 30 years worth of experiments for which we have the same set of genotypes. All those black dots is each hybrid released or commercialized in each year for 30 years. So that experiment was run 30 years in 3 locations in Iowa. And because the plant population, think about that logistic map and chaos, you push the density too high, the old hybrid crashes, you put too low, the new hybrids don't work as well. The same experiment have now a management dimension too, normal, high, and low densities. So for any year in this lattice, what we can do is you can plot grain yield, the year of commercialization, and in this example, you have a normal density for the year 2000, you can calculate the rate of genetic gain. And we can do the same thing. We can calculate all these slopes, which are the genetic gains for each year under low density, normal density, and high density. And this is what I'm showing here. And remember that they're confusing because the axis is year of commercialization to calculate genetic gain and year of when the experiment was conducted in this lower panel. So don't get confused on that. So for the lower panel, think about this panel L, 1990 to 2020, think about the rate of genetic gain is about 2.5, pretty steady. So think about that. We can call it G as your rate of genetic gain. Certain genotypes contribute to maintain increased yields over time. So if you increase the plant population, and let's assume for the moment that there's no trend in there, just by adding a management dimension, the interaction enables the breeders to triple about that rate of genetic gain. Essentially, it tells you that the modern hybrids can tolerate better the density, the rate of genetic gain could be higher. If you increase the plant population even further, now you start talking about the genotype by management, by marrow interaction. The rate of genetic gain in the Midwest is accelerating. So let me stop there for a moment before we start thinking about what may be the causes of this finding. One thing is for sure from all these 30 years of worth of data is that the rate of genetic gain were always positive over 30 years of experimentation in the Central Corn Belt. This is the largest crop production, the most productive system in the world. And some question that come to mind, and I scratched my head many times and I gave up already, is why PNAS published an article saying that not genetics is improving yields in favorable environment. I think this is, in my mind, pretty nonsense. We have data, but anyways, it's for you to decide. I already gave up on this one. So what I will do is I'm going to focus on hypotheses of why the genetic gain is accelerating. And this is important to unravel because if this is a generalized trend, then the climate change is not a problem. Actually, climate change is accelerating food production. So we have less of a problem. If this is one exceptional example, is just for corn, then we might have a problem with climate change. I think the big problem is we have very little information to make any decision. And this is why I think we are in a very poor situation in terms of how we are going to breed for the future. So one thing is genetic gain increase with favorable water conditions. You saw the precipitation was increased over time. The other one is that genetic gain increase is due to this long-term adaptation to lower amplitude temperatures. And let me elaborate in these two hypotheses. So first, let's go back to the key process that is driving yield in corn. This is an experiment we ran and published in Journal Experimental Botany. Let's focus on this W1210, less stress, more stress for yield. This lower left panel, black bars are the double cross. Think about the old hybrids. The white bars or open bars are for the single cross or modern hybrids. So you can see is regardless of the environment, the water environment, modern hybrids yield more than the old ones, and the more severe the stress, the higher the delta. So you have heterogeneity of variances in here, if you look at it in a G/E perspective. So what happens is you increase the stress, all hybrids crush more, they express more barrenness, scatter grain, and reduce kernel numbers. So what happens, or what would happen is if the water conditions are improving in the Corn Belt, essentially you have at the same time an improvement in the environment and improvement in genetics. So something that may happen here is that instead of accelerating, this will predict that the rate of genetic gain should be a little bit collapsing, if anything. So again, well, what can we estimate or have a sense for how big or which is the magnitude of the change? There are 100 millimeters of rainfall increase in the Corn Belt. It's a low amount of water. It's about 30% of dryland conditions. So we can use this theoretical study we conduct with Mark Cooper to estimate if the average evapotranspiration in the Corn Belt is about 500 millimeters, you add another 100, you can see a huge impact, at least when you look at this front, which is the best managed crops that you can deliver on the farm. So this hypothesis, if anything, is going against of what we think is accelerating. The alternative hypothesis is looking at light use efficiency. There's evidence, another paper that we're waiting for, hopefully get published soon, that selection or for yield increased radiation use efficiency about 30% in 100 years of breeding. The other thing we know is that the light use efficiency is highly correlated with the amplitude temperature, which is the one that's been decreasing steadily over the last 100 years. So the argument here is breeders has been improving radiation use efficiency. Hypothesis is that it may be an improved adaptation to this decreasing amplitude. And because this has been happening for 100 years, what happened in, let's say, from the 20s to the 90s was really predictive of what happened in the 90s to the 30s, because it's over the same environmental trajectory. So in this particular case, the environment was decreasing, genetic is going increasing, and hopefully we don't stay on the same place. But I hope you can see the argument here is what we've been selecting in the past, it was predictive of the environment that were coming in the future, and therefore, when you have that agreement, the correlation of performance of the old genotypes in new environments, you expect to be high. So this, we get to the end of the presentation, and call it chapter 10, because it's been published in 1996 in a wonderful chapter by Mark Cooper, which is about breeding G by E and essentially the correlation, or what you expect in terms of correlation of covariance between the selection environment and your target population of environment. So here you can map into these four corners what may happen when G by E is present in the multi-environment trials and the TPE. So think about the multi-environment trials, maybe start on the left side or left peak on this plane, orange plane. Look at the MET for 2020 and TPE for 2020. That's our current breeding system. We breed for our current environment, so there's a positive covariance, right? Go to the opposite side. If you think, okay, we're going to breed for climate change, for 2050 multi-environment trials, of course, you're going to have a high correlation if we get this right. Think about this corner. If you were in 2030, the multi-environment trials that were conducted in the 20 to the, let's say, 60s or 90s were predictive of the 2020. Therefore, you have a high correlation between the two, and that may explain why the genetic gain is maintained or accelerated. The key problem here is, what if these situations start changing? What happens if we run multi-environment trials mimicking 2050 environment? Essentially, we start breeding for climate change today, but we test those hybrids in 2020 environments. If the correlation is not good, we are going to get too ahead of us, and essentially are going to starve today's populations. The opposite, if you don't start breeding for climate change, you're going to starve future populations. The question is where we are at. In the US Corn Belt example, which is the only experiment I'm aware of, we can say we are somewhere in there. Essentially, the pace of environment change, if it's tied to the changes in amplitude temperature, you can expect to continue that rate of genetic gain, and you expect to continue perhaps the acceleration. So now the question is, what was going to happen with wheat? Do we have information? Maybe there's one experiment, maybe in Europe, G by E in wheat is huge. What's going to happen with rice, other major crop, maybe minor crops? Where do we stand in terms of breeding for these changing climates? I will leave that as an open question, and hopefully you will agree that yes, we can think about agriculture as a solution to climate change, but we desperately need a framework for predicting for climate change to inform policy and decisions. I think I will leave it with this nice cartoon that I think we need to start taking action pretty soon. Thank you very much.
Any questions from the audience? I know some students might have gone to other classes, but yeah, any questions in the room? Louis.
Do you hear me?
Yes.
Yes, okay, good. I was just wondering, at the very end of your presentation, you were talking about this chapter, the vision 1996, and I was wondering, so the breeding techniques and technologies have evolved a lot since 1996, and to what extent we should devote a lot of energy for breeding for the future if we don't know yet, maybe in the future the technologies will be so advanced that we will be able to breed much faster, and we should rather dedicate more energy to this. So I'm just wondering, what is your take on that?
I think the first bit that I think is missing is to start monitoring or have a sense for where we are at with different crops as we have that intuition for maize. I think the answers are going to be very different in various regions, regardless of what technology. You can think about gene editing. It may or may not be the answer. Maybe we don't need to do anything. I think the lack of where we are at that we need to solve. That to me is the first question, is develop that intuition for what's happening with the other crops. The second is understanding in corn, is this acceleration will continue or is going to stop at some point in time? I think we are approximating yield potentials. I think this quantitative genetics-based breeding is extremely effective. So if we can use AI and some of the physiological models, it will help us inform what can we do in terms of breeding, and it can accelerate genetic gain. What are the other technologies? Frankly, maybe we need to start thinking of what technology. I don't think changing one gene at a time is going to do it. There's like, what, 20 or 30 years of biotechnology that didn't deliver on that ambition. So I will focus my efforts on building upon the framework that works, which is the quantitative genetics.
Other questions from the room? Tim?
I'm wondering, with respect to questions like climate change, but just in general, to what extent can we hope that with improved breeding, we could design plants that have a better ability to have plasticity in the way they respond or acclimate to various environments, and in that way decrease the G by E? In other words, we know plants already have an enormous capability of changing what they do depending on the environment that they sense at a particular moment or a period of time. Could there be opportunities to further improve that kind of favorable acclimation or plasticity?
Oh, absolutely. I think the best example is coming from Africa, actually, where the selection for plasticity in tillering and prolificacy actually is leading to yield stability. Part of the yield stability is being part of the criteria in maize breeding over the last century, and that's why you don't see that much G by E, in my opinion. Recent studies on tillering, it suggests, contrary to what we thought, that will be an yield drag penalty, is actually not compromising the yield potential, at least in the environments that were tested in Kansas, and we are seeing exactly the same thing in Africa, at least in South Africa, in pretty severe stress environment. So the breeders in these marginal regions improve that, if you will, kernel set, and the resilient or created this plastic corn, if you will, people don't like to see the tillers, but based on our latest research, it doesn't seem to hurt. And when it rains, they can adapt to that better environment. So I think you have, my mind, pretty compelling evidence from that tillering and prolificacy that is possible to use molecular breeding to accelerate and breed for that plasticity. I think the other question, you need to look at the traits that you're going to manipulate. For example, the conductance to humidity, by and large, is not having to have a lot of G by E because it's going to compensate. But if you push it to certain extremes, you may see the G by E interaction. So I think it's totally possible.
Great, thank you very much.
Thank you. One more question in the room before we go to the Zoom audience. Yes.
So thank you very much for a great presentation. Just to ask more about some traits that are capable of maintaining productivity in different climate change scenarios. Here in the Northeast, we're the region in the continental US with the greatest increase in extreme precipitation events. So when I think about how climate change is going to affect agriculture in the Northeast, I think it's not only just going to increase precipitation, but the extreme events of precipitation. So are there any plant traits that you think of when breeding that are able to maintain productivity, not with just more precipitation, but more extreme precipitation?
I don't have an answer to that. If I need to answer that question, I will use the models perhaps Yin is using to develop an intuition what may be the trait. I think that will be a really interesting example, not only to maintain productivity, but with this extreme event, how you're going to prevent leaching and environmental externalities. So I would say you have maybe a more interesting puzzle on how to do both at the same time. But I think these are complex systems. The type of studies we did for this conductance response to VPD, I will use simulation to explore, develop an intuition for what are the traits that may be useful.
Thank you. So 3 questions from Zoom audience. I saw Carla and her student Joshua Fan, they raised their hands first, and student. Yeah, so Carla, please go ahead.
Oh, hi, Charlie. Fantastic talk and very exciting. And I see Joshua also has a question, so I'm going to be brief because probably he will have a question along the same lines. I'm very intrigued and excited to see that you are, combining knowledge in addition to data. In fact, I'm a big fan of that approach. I think that the data-driven approach that everybody is using nowadays is not going to take us that far. And indeed, a much more meaningful way is combining, knowledge with the data and incorporating as much prior knowledge as possible. So this is a bit my question, basically, and this is maybe a long question, so we'll talk at another time. But it sounds that you are using these mechanistic models, and maybe the main way for the way you are incorporating this prior knowledge is by generating data with the models and then fitting some kind of model to predict. Is that what you are doing?
We are calculating some deltas using this approximate Bayesian computation. Not really fitting a model, but we fit the marker within the. We are doing the fitting of markers within the dynamical model. That's
yeah. And using this Bayesian
it's an optimization. Yes.
Optimization reasoning. Okay. Yeah. Very good. I would like to learn more, and maybe we can talk another time. And thanks so much again. Great talk.
what I really would like to do is try your Sudoku algorithm and see how we can put the crop model within that framework.
Yeah. So yeah, I would like to think about it. Indeed, I mean, the key part is how we can develop. Typically, neural networks have all these layers that are black boxes, and we like, using interpretable latent space so that we can, condition the model better based on prior knowledge. Yeah, and thanks. I love the Sudoku analogy because I think that's easier to understand than material science. So thank you for mentioning it.
Joshua, do you have any follow-up questions along the same line, Carla?
Oh yeah, sure. So I guess I'm Carla's student, and I was also just wondering about for these crop models, do they generally have a way to incorporate uncertainty, such as like uncertainty about the future weather or uncertainty about our knowledge of the processes? Like there might be some processes that are not modeled.
You can actually, if you want to come to Florida, I would love to work with you on that. Yes, you can. We can write it in a way that you can embed uncertainty in the parameters. We can put functions and again, a hypothetical connection in the network, and then you can make it kind of fuzzy and ask the question whether that process exists or to what extent exists. And the future weather, yes, that's been done pretty routinely.
Sorry, a follow-up. Is this leveraging on this Bayesian framework?
Yeah. And
Cool, thank you. I'll definitely take a look and try to learn more about this.
yeah, send me an email. I'm more than happy to talk with you. I've been trying to do that, but I need a smart student like you and a professor like Carla to get it done.
Thank you.
Okay, thanks, Charlie. It was fantastic to have that talk, and it winds me back around thinking about rice. And so my question for you is that the question, and then I'll explain it. The question is, do you think that hybrids convey some kind of advantage in the face of the challenges that we face in the breeding community? And the reason I'm asking is because, as you know very well, rice is normally bred as an inbred, except in China, because heterosis is not that big an advantage for us. However, in rain-fed environments where we don't manage the water, it seems that the hybrids are sometimes more resilient to the variation that we have in both hydraulics and water and temperature and other things. And so the grain quality suffers a bit because there's more variance, okay? So for the discriminating eater, the quality isn't there, but for simply providing quality carbohydrates, the hybrid might be a good solution. But I'm asking now specifically in the context of climate change, do you have any reason to believe, or do you have any priors on this to suggest that the hybrid route might be better than the inbred route for rice?
Not a rice expert by any means, but I can borrow information from what happened in maize breeding. And if you look at the females inbred, they look like hybrids. So unless you have a compelling evidence that heterosis is a major driver of performance, you may get away and make faster progress just breeding the inbred type. In wheat is the other thing, right? How many years they've been trying to form heterotic groups, and I haven't seen a lot of that is really compelling. So
there's no complementation in the context of the pathways and the manipulations of prior knowledge that we know that would provide an advantage for having two alleles rather than one at these loci.
Again, I'm thinking about corn, where there's some even genomic regions missing that perhaps is a big part of the heterosis in maize. I don't know in rice.
Anyway, I'll leave it as a question. I think it's one that for the rice community is quite compelling because the changes that we have to breed for include emerging from the irrigated system
Mhm.
with very, very different root structures and all kinds of things. So it'd be fun to see what the parallels will bring. I remember a conversation with Dubik many years ago, and he said, once we get rid of the deleterious recessive, we're gonna be modeling just like you guys, meaning in inbreds. So maybe your answer sort of reflects that.
Yeah, it makes sense.
Thanks.
A comment from Ed Butler's group. How much does inbred G by E correlate with hybrid G by E?
Inbred G by E, I don't know. All my experience was in hybrids, and every time we tried to predict hybrid from inbreds was not a very successful endeavor in maize.
Yeah.
I don't know, Ed.
Yeah, Charlie, I think that's going back to Susan's question. That I think, yes, I agree. Those inbreds look great, but I only ever see those inbreds under good environmental conditions.
True.
If we put them under the same G by E stress, I wonder if we'd see something different, and that there is environmental range buffering there, and for wheat people out there, this is, I think, with the D. There's an argument the D genome is that environmental buffer.
Oh, okay.
I think it's something we should be asking, and we could ask it using the maize inbreds, but we haven't probably. Okay, thanks.
Maybe the question for you, Susan and Ed, and going backwards, I think we need a framework and start organizing these views for breeding for climate change, because otherwise we end up with what is the idiotype rather than how we are going to breed today for tomorrow for 2030 without starving populations until we get the final answer. And from what I've been reading is, I don't think breeders were involved enough to define that path. So if you're interested, I would like to spend more time on that. And I think the AI with Carla is part of the means to get to that accelerated breeding.
I think it's definitely worth conversations, and it might be worth a panel or a some kind of workshop.
Yeah.
Just to list the questions and organize our thinking. If we could combine inbred and hybrid, species, if you will, and breeding approaches, I think it's kind of an important opportunity. Because I even hear, of course, this, potatoes going forward to breed hybrids based on inbred diploids. So people are doing somersaults in the air to try to come down on their feet these days and willing to try anything.
Yeah, I hear potato is not the fastest. They were growing potatoes from what, the 30s? At least in Florida.
Thank you very much for everyone joining the seminar today.
This has been a production of Cornell University, on the web at cornell.edu.